All right. Well, we are going to start on doing docking now, and do not worry if you could not get some of the files or some things to run from the last notebook. We're going to start from already prepared files that we're going to be downloading. So even if you weren't able to follow along with the previous one, if there were some parts that didn't work, you should still be able to follow along with the docking notebook. Okay, so let me share my screen. You can either click on our next notebook from the previous one. So, if you scroll back up to the top and click on number 4, molecular docking using Nina, then you can open the notebook. Or if you have this Google Doc guide, you can click on it from there. And then would someone mind like maybe spotlighting me? Oh wait, I got it. Okay, I'm spotlighting myself. So you can open it from there. This is the notebook. It's 04 Cheminfo Crash Course. And then you can do the same thing that we've been doing to save a copy for yourself. I noticed that there's also this copy to drive button, so that probably works too. So let's just go to File and then save a copy in Drive. Okay, and then now this is something that you can save for yourself as we do this. Okay, so let's scroll past this at the beginning and start our notebook on installing some of our packages, and then we'll go back and talk about this information. So go down to your setup section and run this cell for setup. So this is going to install some of the packages that we're going to be using. We're going to be using Pat's package, Useful RDKit Utils, Hi3DMOL, which is the same visualization program that Levi just used, and then also RDKit. And then we're also installing OpenBabel. So press this cell to run. And that will run for a little while. And then we are also going to need to download our docking program. We're going to be docking with an executable named Nina, and we can download that directly from GitHub. So you can go ahead and start this running as well, and that's going to start your download. And I really hope that we can all download this at the same time without overwhelming GitHub. That would not be good. And then this is something to make Nina executable. You can run that as well, and that will like complete the setup that we need to do. So mine is downloading, so that's a good thing. Hopefully everyone's getting their executable downloaded. So since we ran that, I'll just scroll up a little bit to the top and talk about what we are going to be doing in this notebook. So we are going to be running molecular docking on those ligands that we prepared previously. So Pat showed us how we mined those from BindingDB. Levi showed us how we can get 3D structures for everything for docking. And now we're actually going to perform the docking calculation. We're going to use a program called Nina for docking. And if you click on this link, that will take you to the Nina GitHub. But Nina, it's spelled G-N-I-N-A, but it's pronounced Nina with a silent G. It's a fork of a software program called Smina, and Smina is a fork of AutoDocVina. So if you were in our workshop last year, we used AutoDocVina to do our docking. Out of AutoDocVina, and for those who are unfamiliar with software development lingo, a fork refers to a copy of a software package that can kind of be developed almost in parallel with the other one. So it will fork off at a particular point, and then it can also continue to be developed. But Smina was a fork from AutoDocVina that was created to allow easier setup of docking calculations. So it allows you to do things like define a binding site automatically based on a ligand. And there's also, you can also have more customization of the scoring function. So for AutoDocVina, there are a particular set of scoring functions that are built into docking. But for both Smina and Nina, that's more customizable. And then Nina, in contrast with Smina, so we have 3 different ones that we're talking about. Nina adds a step on the ligand scoring of a convolutional or some convolutional neural networks that can be used to also like score and rank your ligands. so rank your ligand poses. So that's the difference between SMINA and NINA, is the addition of the neural network for ranking ligand poses. So because we are going to be running NINA, which requires doing machine learning, it uses PyTorch underneath, it is very beneficial to run this with a GPU. This notebook should be set up to run with a T4 GPU. So if we click on with this on Google CoLab, you can see that I have Google CoLab Pro, so I could use a different kind of GPU if I wanted to, but the T4 is the one that is free. If you wanted to get Google CoLab Pro, you can change the runtime type, and then you can pick one. And this actually will run, I'll just go ahead and tell you guys, it will run significantly faster on either the L4 GPU or the A100, if you have that available. So that's what we're going to do today. We're going to start out by taking that crystal structure from the PDB. We're going to take the ligand that was in it originally. We're going to redock that and see how well NINA predicts the crystal ligand pose. We're going to do something called cross-docking, and that is where you take a different PDB. It's the same protein structure, but a different PDB ID, and dock the ligand to that and see how well it does with that. And that's going to kind of give us an idea of how good our docking, how well our docking program is able to reproduce experimental structures. Then we're going to dock all of those ligands that we prepared. So that's kind of where we are going today. Okay, so we should have everything set up. The next thing we're going to do, I mentioned that we have all of the docking files prepared already. So we have those prepared and organized. We're also going to download a utility function for visualization. So you can run this cell, and that's going to download those two files for you. If I click over here, I'll now see that I've downloaded a file called dockingfile.zip and then util.py. And I see that I had a request to zoom in, so I'm zooming in. And then we should unzip that docking file so you can run that. And you can see that that is going to unzip that file. And here we have the 3D structures sort of that we prepared in the previous notebook, but I've organized them a little bit. So we have within the docking files folder, we have another subdirectory for protein structures, and this contains different protein structures that we're going to use for docking. And we have ligand structures that we are going to be using for docking. Okay, so that is our setup. Let's move on to docking with NINA. So I've been talking a lot about docking. I'll say when you dock, there are two different steps to docking. There is sampling and then scoring. So you'll take a 3D structure of a protein and define your binding site. You'll take a 3D structure of a ligand, and then the docking program will sample the binding site with that ligand. It will score those, and then it will rank them on the score. And that's generally how docking works. Okay, so the first thing that we're going to do is we are going to redock. So we used a protein structure called 7-LME. That had a ligand bound called Y6J. And the first thing that we're going to do is we're going to take that, we have taken that ligand out, and we are going to use the docking program to kind of put it back into the binding site and see if the docking program predicts the correct pose. This first cell is just going to run a visualization for you. So this is the ligand that is in the PDB structure. That is in the PDB structure. We did some in the previous notebook. We made sure that the bond orders were correct here, but we can see where this is bound. And if you kind of zoom out, you can see the full structure. And then after we view that, we want to actually run our docking calculation. I want to get us started on the docking calculation, and then we'll come up and talk about the different parameters that we've put in. So let's scroll down to this cell. This makes a folder for our results, and then it runs NINA for docking. So go ahead and press to run this. That's going to start running it. And in the few minutes that it takes to do our docking of this ligand, let's take a moment and talk about what this command is and what the different options mean. Okay, so this is an explanation of Nina. So here, when we run Nina, we specify the program that we're running. Then we have an argument that is -R. The R here is for our receptor. We're setting our receptor to look in our docking files folder and then at this structure 7LME. I think this isn't quite up to date, but it's just 7lme.pdbqt, which was the pdbqt file we prepared previously. We use -L and then give it the location of the ligand that we are docking. When we are docking with both Mina and Nina, we can define the docking search box automatically by using an argument called AutoBoxLigand. We are using the known experimental pose for this. So we do --AutoBoxLigand, and then we give the location of that ligand file. Next, we specify the output file path where the resulting dock ligand will be saved. We are going to save all of our docking results in a folder called docking results. And then it's called Y6J docked. So, and then because the sampling algorithm for docking, it's a Monte Carlo chain. If we want our docking to be reproducible, in other words, if we want to get the same answer every time, which reproducibility is a good thing here, we want to set our seed. So you can set the seed to any value. We're just going to use 0, but this using a fixed seed is going to make our docking reproducible. Then we can set the exhaustiveness level. This corresponds to how many different Monte Carlo chains you're running for the ligand. The default is 8. And I played around with numbers a little bit, and I found that for the system, a good exhaustiveness level was 16. So that is what we are going with. So that is, we scroll down. We couldn't put that in line with the command because this is a bash command, but those are all of the inputs or all of the settings that we've set for this docking calculation. So let's scroll down and see how we're doing. We can see that this is running. If you still have time, you may try these different mouse inputs for Pi3D-Mol for the visualization. Okay. And then we have someone, so I see someone said, someone asked, how do we get the pose of the ligand saved in an SDF format? This -O command that we used is where we're specifying the output file name. So this will make it so that our poses are saved in SDF. So here is the output from Nina. Mine has now finished running. It took about 3 minutes. And then we have some information here on the calculated affinity, the CNN post score, the CNN affinity. So we want to work on interpreting this output. I'll also note that these calculated values are stored in that SDF file that we wrote out in this docking results, and we can use RDKit to pull those out, and we'll be seeing that later in the notebook. Okay, so interpreting the output. So when it finishes a docking run, it prints a summary table for the generated poses. The columns correspond to the following. So we have the mode, which is the pose rank. We have the affinity, and this affinity comes from the AutoDoc-Vina score. And yeah, so this is really just what comes out of AutoDoc-Vina usually. This is some kind of intramolar strain energy according to the Vina function. And then Nina adds on top of this a CNN pose score. So this is from the convolutional neural network, where higher values closer to 1 indicate that the neural network is more confident in this geometry. And then we have the CNN affinity, which is the CNN's prediction of the binding affinity, and this is expressed in PK units. So here for the CNN affinity, a higher value means stronger binding, whereas for the Venus score, it's the opposite. We want the more negative values for stronger binding. And so when we look at the scores for the ligands, so Nina is going to rank these, I believe, on the CNN pose score. And it's going to say like our top one is based on this column. So we can visualize these docked structures. So what we're doing right now is we're trying to figure out if overall, if Nina does a good job of predicting what the structure will be. You'll have to remember that even though we've seen how the ligand fits into this particular binding site, Nina actually doesn't know about that. So when we do this redocking, we're able to see how closely Nina is able to reproduce the experimentally determined binding structure. And so in this structure, what we're looking at, the yellow one is the ligand from the crystal structure, or sometimes called the cognate. And then the green molecule here is what Nina predicted the pose to be. And just visually, this looks very close. But one way we can measure this is by using the root mean square deviation. And if you've worked with molecular dynamics or other types of atomistic simulations, you may have seen this before. The root mean square deviation is just a measurement of how the coordinates of two different structures differ from one another. And we're going to use from Pat's package, useful RD-Kit utils, we're going to be using an RMSD for maximum common substructure. For our case, when we're doing this redocking, the ligand from the crystal structure matches exactly the ligand that we are docking, so it's going to be a perfect match. But the nice thing about using this maximum common substructure RMSD is that we can use it to kind of compare how similarly slightly different ligands bind in the binding site. So let's run this one. And I'll say that when we're doing redocking, we're evaluating how well the docking program predicts an experimental structure. What's usually considered a good redocking is if our measured RMSD is below 2 angstroms. So that's kind of what we'd be looking for here for our top pose. And so this is taking a moment to run. A little bit longer than usual, actually. Okay, and so we do see that when we do that RMSD, our measured RMSD is below 2, so we would consider this docking to be successful. Okay, so let's take a few minutes here. Yeah, let's take a minute or two here for the exercise. So what I would like you to do is try docking yourself. Try turning off the rescoring with the convolutional neural network. You can do this by adding CNN scoring none to your Nina command. And then if you do this, make sure you save your results in a new file. And then let's see if using the CNN affects, how well our top pose predicts the crystal structure. So I'll pause here for a minute or so that you can try that out. And if you get an answer, feel free to send it to the chat. I'm actually going to go ahead and fill in the code here. So you could just copy what we had before and then add another line here, CNN scoring none. So that's going to turn off the rescoring. So I'm going to go ahead and run that. And I believe it does run more quickly. Oh, and make sure that when you run it that you do change your output docking results file name. And if you added CNN scoring none and it's not working, make sure that you have this little slash here, or it will not run. This is a syntax that we're using that allows you to have a multi-line bash command. So we have a question on how the docking score correlates to the binding energy. And I would say that based on my experience, is highly variable, depending on various things. I think you would hope to get a good score, but it's not necessarily guaranteed. Okay, so we have a question on the docking algorithm, and I wish I had an illustration of it for you, but what happens is right now we're doing something called rigid docking. We have our protein structure or our receptor structure, and then we have our ligand that we want to fit into some kind of pocket on the receptor. We've defined here a box. So we defined a binding site using, so the binding site that we've defined, if we scroll up here, is based on where this ligand is. So we're saying, and what Nina does automatically is it will take this ligand, it will put a box around it, and then it adds a buffer. I think it's about 4 angstroms. And then it also kind of, I think if your ligand is kind of rectangular, it makes sure that it's more cubic. But what happens is we take another ligand, we'll kind of fit it into this box. The algorithm will try different configurations or different conformations of this ligand. And then for each one, it's going to give it a score. And this score is can be based on different things. We're using the default Nina score, but it's going to kind of be based on where those atoms are and interactions with your receptor. As it goes through, it's going to try lots and lots of these. It's going to give them a score, and then it's going to keep the best one. And then at the end, it will give you back the structures that kind of give you the best scores. And that's how the docking, hopefully that answers some questions, few more questions about how the docking algorithm works. Okay, so we can check if our exercise is done. So mine is done. You'll notice that when we're running this without the CNN rescoring, our CNN post score is all -1 and our CNN affinity is 0. And then if we run our RMSD here, it's actually very interesting. We get different pose rankings if we're using the CNN or if we're not. And what Vina predicts the top pose to be is about 8 angstroms different, or has an RMSD of about 8, which is very different from the crystal structure. So that is interesting. I think I didn't quite explain, let's say when we use Nina, the default for Nina is that it does the, it only uses the convolutional neural network at the end. So it will run the docking similar to how it's run in Vina and Smina, and it gets kind of maybe something like 50 top poses. And then when you're using Nina, it will reorder those using the convolutional neural network. And so it really prioritizes things that it thinks are more geometrically accurate. So depending on whether you are using Nina or Vina or Smina, your poses may be ordered differently. Okay, so we can also do this thing called cross-docking that you'll see done. So what we see here is that Nina does a great job of fitting the ligand back into its original structure. However, this is kind of a best case scenario. It's a kind of an easy problem because we're fitting a ligand back into the exact place where it was. So this structure is kind of perfectly formed for the ligand when we're doing redocking. Another thing that you'll see done to evaluate is cross-docking, and that's when you take a different PDB structure. We're going to look at the one 7L11. So this is also the main protease, but it's a different crystal structure of it, and we can also dock our ligand to that, so we can view this if we'd like. This had a different ligand bound. This one had XF1. And let's start running that. This is also going to take 3 minutes, but don't worry, this is the last one that we are running. The rest of them we're going to load in pre-computed poses. But we may want to do cross-docking because what we should expect is that our ligand should bind to this structure in a similar way as the other one. I'll just also say here, if you're doing cross-docking and you want to measure the RMSD, you need your protein backbones to be aligned with one another. If you don't, and you measure the RMSD of the ligands, you're going to mostly get a result that is based on the translation and orientation of the protein. I think the easiest way to align protein structures is to use VMD. Particularly the RMSD tool. There's an RMSD calculator tool where you can align selection. So that would be my advice. While we are waiting, are there any questions? If anyone has seen any interesting ones from the Discord.
One question was, how is the CNN actually used in the docking scoring if it's after the fact, especially?
Right. So with Nina, you can use the CNN in different ways. They suggest using it for the default, which is a rescoring of the ligands. But you can also use the CNN throughout docking. So what the CNN does is it takes the structure and it assigns a score to it based on sort of its training. You can use the CNN throughout the docking process. That is much, much slower and much, much more computationally expensive and typically doesn't provide better results. The way the CNN is used here is just at the end to reorder the poses that you have. You can also use, like, there's also a setting for refinement that uses it kind of a step before the reordering that is also more computationally expensive. That is typically not recommended. So the way the CNN is used is that it's just used at the end of the docking to reorder the poses. And so cross-docking, someone asked for clarification on what cross-docking is. Cross-docking is using the same ligand on its protein structure, but like a different version of the protein structure. Because we would expect that it will bind to the main protease similarly. Okay. All right, and these are our results, and we can measure the RMSD here. Again, we get good results for this cross-docking, meaning that Nina is doing a good job predicting the binding pose of this like native ligand. Okay, so we can visualize this again to see the difference. If you'd like to see other poses that were written out, we can change this animate to true. And this is going to just play through the different poses that were predicted. So you can see an animation of that. Okay. So I see we have a question in the chat on how you can download these scores. These scores are all stored in the SDF file, and we can pull those out using RDKit, and we're going to see that in this next section. Okay, so if we want to dock multiple ligands, so in the previous notebooks, we pulled out multiple ligands from binding DB. And then we also prepared a set of those as well. If we have all of those in one SDF, we can use Nina to dock those all at once. When we do docking with many ligands, we recommend those to be in all one SDF file. And then we recommend, or it is recommended to use a PDB-QT for the protein. Because this takes, as you can imagine, we have something like 6 ligands to dock, and just based on the docking that we've done so far, you can probably imagine that it would take a long time to run all of these. These have been pre-computed. We'll retrieve them from the repository and then unzip them. You should now see several files in your docking results folder. So I think when I ran it, it took something about 20 minutes. And so if you were to run this docking command, you could wait several minutes for this, about 20, and then you would get this SDF file called multiple ligands docked.sdf. I do want to see if I can show you what this looks like, because there have been questions about where and how we can get docking results. But if we were to, oh, actually, I have it here, I think. So let's actually take a look at what's in this file. So I am just uncommenting this. This is a command that we can use to preview what is in that file. Oh, multiple ligands. I changed the file name, but there. So we have to change this file name, but we can preview what is in this file. And if we look at this, this is an SDF. This is what comes from Nina. It's an SDF of all of our docked poses. You'll see that here is the information that we had in that we saw that was printed from Nina. So if you had a question of where you can find the information that was in the table, it's stored directly in this SDF. So I'm going to comment that back out. And we're going to use RDKit pandas tools to pull that information from the file and get it into a data frame for us to work with further. So the score columns are like a minimized affinity, a CNN score, the CNN affinity. There's a CNN VS score, and that's something where we've multiplied the CNN score and the affinity, and then the variance. So we're defining here what our score columns are. Then we are using, we have these different SDFs. So we're going to take our sort of native ligand structure, and then we can use pandas tools, load SDF to pull all of that information from the SDF into a data frame. We can concatenate those two together, and then we're going to have a data frame that contains all of our data and our scores from docking. So we have here the compound ID, So we have all those different compound IDs, and then also here the native ligand at the bottom. And then we can use pandas functions to sort by different metrics in our different scoring metrics. So for example, if we want to see the top pose for each one ranked by minimized affinity, we can fill in minimized affinity here. So this minimized affinity would be our affinity based just on the NINA score. We can also, we could change this and look at sort of what compounds had the top CNN score. Let's see, and so we would want to change ascending to false. For the affinity, we typically want to see the most negative value. For these other ones, we want the most positive value. Okay, so we can kind of use this to filter our data frame in different ways to see which compounds have higher scores for particular metrics. One thing that's interesting about these results is that our minimized affinity, the order that we see, we actually want to change this to true. It correlates pretty well with our IC50 values. And even though that's true, I would kind of like throw a caveat out there that we may not be able to conclude much from this because we'd really want to do kind of more docking. And perhaps docking with different structures to make sure that this is true. Okay. So this is somewhat interesting that when we rank by our minimized affinity, it kind of matches our IC50 values. But again, what you choose for your score is going to kind of depend on what you're looking at. Okay, and then we can also use this visualize poses to look at how these different the different ligands bind with our receptor. So this is going to kind of play through all of our ligands and all of the poses. Here, we can pick one particular compound to examine and sort of see how much differently it binds by RMSD. Okay, and then, yeah, so we can visualize that as well. So we just saw docking with NINA. We did rigid protein docking with a set of ligands. We pulled those results out into a data frame, and then we kind of filtered on different metrics for that. NINA has options for doing lots of different types of docking. So you could define your own scoring function. You could kind of set more information. There are different CNN models available in NINA that you could set. Also with NINA, you can do flexible docking, where you allow specific receptor side chains to be treated as flexible. You can use arguments like flexdist ligand to specify a reference point, or flexdist to set the distance threshold. So you can do flexible docking that way. Nina also lets you do something called whole protein docking, and that is when you let the ligand explore the surface, the full surface of the protein. And that can be used if the binding site isn't known beforehand. I would expect that. I haven't tried it, but I would expect that it's very computationally expensive. And what you do if you want to do whole protein docking is you'll set autobox ligand to be equal to the receptor itself, and that will allow the ligand to go across the surface of the protein. If you are doing whole protein docking, it's recommended to set the exhaustiveness a lot higher. And there's some, I think, additional guidelines for that on the Nina GitHub. And however, it's really advised to be careful with these options as they're much more computationally expensive, as in they'll take a much longer time to run than using rigid docking with a known binding site. So throughout this tutorial, oh, I know I was missing my visualization. So throughout this tutorial, we've performed docking calculations, treating the protein as a rigid receptor. This is a really common thing to do, and it's very computationally efficient. And I think especially you've seen for this workshop, we wouldn't want to be waiting much longer. However, there are times when the binding site might need to adjust its shape to accommodate a ligand, and this is called like induced fit. So induced fit is actually something that is observed the main protease. We're probably getting very good results here on redocking because our ligand sites are already, they have a good shape for these non-covalent molecules that we're docking. But if we were starting from some kind of structure, maybe if we were starting from a structure that didn't have any ligand bound, we may not get as good of results. So according to this paper that we started from in the beginning, the binding site of the main protease is found to be flexible. Particularly, it highlights two residues, 189 and 49, that have flexible side chains that are near the binding site. So actually, Nina will allow you to kind of define flexible residues if you would like. So we can specifically define these residues to be flexible for docking. So I meant to show, actually want to see if I can find, I meant to show here a visualization of those residues in the binding site, but I see that it's missing. So let me see if I have it somewhere. Okay. I can paste this code snippet to the Discord. But I will show. So these are residues that are thought to be flexible. There may be other ones in the binding site as well, but we can do flexible docking with Nina also. So for kind of your final challenge, I want you to try out flexible docking. You can either use a cutoff distance with a target using flex ligand and flex distance, or if you have a specific idea of what residues are flexible, you can use this argument called FlexRes. So here it kind of tells you how to try that out. And then try redocking or cross-docking using flexible docking. You'll see that that will take much longer to run. And then challenge 2, which will take longer than we have today, is to try docking all of your ligands to this other 7L11 structure and see if you observe different ordering when you look at the scoring. Okay, so that is what I have for docking with Nina. I think we have some kind of time for Q&A on docking or any other topics from today.
Yeah, we've got a few minutes. Go ahead.
So someone asked if the ligand was already in the PDB, what was the point of ligand screening? So we've docked when we did our last docking set. Which the command is given here. We're docking about 6 different ligands. All of these ligands do not have experimentally determined structures. There is one ligand that we were working with already. There's a PDB structure with a particular ligand, but these ones are not really known. So the first thing that we did was we redocked that ligand to see how good our docking algorithm did on predicting that experimental structure. And then we docked some ligands that we do not know the structure of. Okay, we have a question. Can we do docking with a new molecule with smiles? You can definitely do that, because what we actually started from in Levi's notebook is we had just smiles of these ligands. What happened is we took the smile, we ran mole scrub on them. Mole scrub gave us a 3D structure, and then we docked that 3D structure. So if you were to follow the same procedure, you would take a smiles, you would generate a 3D structure, and then you would use this workflow to do docking. So I see maybe, does anyone else here, like Pat or Levi or Paul, because Pat, I see a question in the chat about screening multiple proteins against the same set of ligands. Would you like to give some thoughts on that?
Yeah, that's an incredibly bad idea.
Okay.
I would never do that. Docking is a crude approximation. And I think it should only really be used when you know the binding site, you know where things are, and then trying to compare docking scores across multiple proteins, you're just asking for trouble.
Okay, and then oh yes, let me share the code to highlight the residues, because I said I would do that.
Oh, can I comment on this one about performing docking on a scale of, what is that, 100 billion or 100 million molecules?
Yes.
Yeah, you need to set up some serious infrastructure to do that. You can't dock, I wouldn't dock more than 100 molecules on CoLab. So, when you start getting into the millions, you really need to set up some serious infrastructure.
I'll say a few, yes, serious infrastructure, and particularly if you're using these T4s, it's very slow. If you can use the L4s or A100s, even that's a little faster.
So there's a question here for if docking is so crude, why does it still have a role or does it still have a role in drug discovery when MD might be more reliable? Sure, MD might be more reliable. I'll start by answering in that docking is very useful because it's significantly faster than MD, especially if you are needing to screen millions, the zinc 15 data set, 10 to the 60 molecules. You're not going to screen that, don't worry. But it gives you an idea of where to look, and then MD you can use to sort of get more refined depending on what you're trying to observe, like you're trying to get like a free energy of binding. And even then, I would say that short MD for that kind of thing is kind of a misnomer, because short MD does not mean that it's a short wall clock amount of time to do. Whereas docking can go through millions of molecules significantly faster than a handful of, quote, short MD simulations.
But can I just add one thing to that?
Go for it.
Yeah, because MD is not going to tell you where a molecule is binding unless you're willing to run tens of microseconds of simulation. And even in that case, it doesn't. So I agree completely with Levi. Docking is crude, but it's a good technique for trying to get some ideas about how a molecule is binding in a particular pocket.
Right, and I'll also say that for a workshop setting, the type of docking that we can do is also very limited. We already saw that, you know, when we're doing docking of like one structure, we're waiting like 3 minutes. Then there's also like flexible docking that you can do, but there's also, that we didn't talk about here, this concept of like ensemble docking. And that is where instead of doing flexible docking, where you allow your side chains to be flexible, you may have several conformations of your target receptor. And those can come from the PDB. Sometimes they come from molecular dynamics simulations also. So in this case, you might run some molecular dynamics simulations, get some different like conformations of your receptors where the binding site is like a little bit different, and run docking with those. And I sent a paper that is from the group that makes Nina about the cache challenge, where they use an approach like that. Just to build on the idea of molecular dynamics and docking.
If the ligand is larger than the pocket, it's just not going to dock.
It seems like we have quite a number of questions here. You know, at this point, we need to officially end our session. But Pat, Jessica, Levi, could you stick around for maybe another 10 minutes to answer a few more of these questions? Are you willing to do that?
Yeah.
Sure, I'd be happy
Yeah,
to.
I can stick around for just a few minutes.
All right, so we're going to leave the questions open for a few more minutes here. And so people, you know, I don't think I can answer any of these, but Jessica, Pat, Levi, take a look and see what you think, because there's like 50 of them in there.
Mm.
I will say one thing for people who are getting ready to leave. You'll get an exit survey by email. Please let us know your thoughts on this session. And also, if you have suggestions for future crash courses you'd like to see, include that in your response to the survey you receive.
Yes, and you will. So Christine just posted the link to it. You will get your exit survey also by email, and it's also where you can get your certification for this course, because I've seen that question come through a few more times.
That's
and then
right.
the videos will be uploaded to the RCSB site, and we will post a reminder here in this Discord when they are available. I don't know exactly when that'll be, but later this summer.
Thanks so much to all of the presenters and for everyone who joined today. It's been really amazing, and appreciate everybody for participating in all the different ways. So thank you so much.
I see someone asking about good coding practices for docking multiple molecules simultaneously. I think for that one, what I would recommend is using that SDF with many ligands, and that you're kind of minimizing the amount of coding that you're doing then. I hope that answers the question.
Yeah, I saw a question before where someone was asking about post-processing the results from lots of docking. One of the things that I like to do is use interaction fingerprints. So there's a package called PROLIF, PROLIF, which is excellent. It generates a fingerprint that characterizes how a molecule binds in a pocket. And then you can use the same sort of techniques I talked about for clustering to then come up with sets of molecules that are binding in the same way. And it makes it easier to visualize things.
So here's an interesting thing about this particular system with PROLIF. PROLIF uses RDKit underneath for processing for the protein And if you, for this particular system, this particular protein, if you load it in ProLive, you run into problems because ProLive is like inferring bonds based on a distance criteria. And I think that it must be something in the binding site. It detects like an incorrect valence. So this particular system at the moment cannot be used with ProLive, but we may file a GitHub issue to see if anything can be done about that.
But it's inferring bonds in the protein structure then.
Yes, because it goes through like the protein. So it's yeah, it's somewhat interesting. That was why Levi and I initially tried a minimization on this structure, which did fix the problem, like, but also made the docking kind of incorrect. I think those flexible residues that we highlighted kind of like closed up.
So there's a question there I just saw is which do you prefer for docking, Gina and Schrodinger, and why? They'll do different things, different ways, and Gina's free.
I remember when I first started reading about docking, the folks who created Vina, Autodock Vina, talked about the idea that their stuff was good plus or minus 2 orders of magnitude or something like that. So it's just an estimate. And I don't know if anybody's claiming better numbers than that these days. That would probably been 15 or 20 years ago. But even now the estimates are still very, very rough. And I'm not sure if the numbers you get from Schrodinger are going to be any better.
The question about how fast is ProLive? It's very fast. You can easily process tens, hundreds, even millions of molecules.
There's a question. Could you suggest a resource to understand theoretical basics of docking and how different programs work?
I would suggest there are several review articles that were written by Brian Schoichet, Schoichet, at UCSF. All of his review articles are excellent.
Can you put that name in the
Sure, I'll put it into Discord.
That'd be great. Thanks very much, Pat. I want to read them myself.
And since I have everyone here, do you all know of any good codes or algorithms that do protein docking well? Because that's been one of the most common questions. We had that last year a lot as well.
One of our guys here at RIT, Emiliano Brini, works with a program called MELD, MELD, which is built on another program, and I don't remember which one it is. But he does protein docking. And maybe what I should do, Levi, is just